Thank you for joining us for this session, Nitrogen-Fixing Corn, Making It a Reality. This afternoon, plant breeder Dr. Walter Goldstein of Mandeman Institute will share his work to breed corn with high protein quality and nitrogen efficiency and nitrogen fixation, and ongoing efforts to test the hybrids on organic farms. Please join me in welcoming Dr. Walter Goldstein.
Yeah, thank you. I wonder how many of you enjoyed the last presentation. Could you raise your hand if you were here for the last one? Oh, just a few of you. Okay. My name is Walter Goldstein. I'm from the Mandeman Institute. It's a small organization in southeastern Wisconsin. We breed corn for organic farmers. We are a nonprofit organization. We have a small company to help us release our seed. And we work together with another seed company who actually produces it and is trying to get it out. And that's called Foundation Organic Seeds. I want to talk with you a little bit about the program that we've had for breeding corn for organic farmers. And I want to talk about nitrogen fixation. And I want to share with you some of the things that we've learned over time in terms of breeding for organic and nitrogen. I think, how many of you know what an inbred is? Okay, well, so when we do breeding, we make crosses, and then we select the plants. We constantly inbreed them in order to fix their traits, and we test them all the time to see if they'll make good hybrids when you cross them with something in what we call a different heterotic pattern. And we find the ones that cross well, and that's what you get when you plant your corn. We also, at our institute, we also breed open pollinateds, but our main emphasis is on producing hybrids. And we're very interested in producing corn that has higher value and is adapted to organic conditions and will take less inputs and produce well. We cross to make a variation using more or less conventional methods. We select the best. We're looking for bodies, whole bodies that perform under organic systems. So we take a bit of a departure from looking at plants as being genetic machinery and a collection of genes or alleles. We look at them as wholes that have to perform and make the right decisions, because growing is a decision-making process by the plant. And we want to find those plants that make the right decisions and do the right things for organic farming under unpredictable conditions. We also have learned that it's not simply a question of genetics, that the plants, as we select them under organic conditions, that they adapt to organic conditions. And so we're very interested in the variation that pops out sometimes as we continue to select under organic, and often under very stressful conditions, because we put our plants through a lot of stress. The shifts that occur in the plants can be unpredictable, and they can be very useful. So we view the plants as partners. We view them as being on the creative end. Often they're rearranging their genome, or they're showing epigenetic variation. New things are popping out. We need to be the partner that's paying attention and making the right decisions and seeing what's coming up and how to utilize it. So we view it as a part that we play the human part, they do the plant part. We better do a good job at doing the human part on the partnership. So that's a little bit our philosophy there. I speak sometimes about emergent evolution, that things happen as we're breeding that we didn't predict. And one of the things that happened that was very important to us, that as we continued to select under conditions where nitrogen was limiting, that we had suddenly the occurrence of a lot of opaque or kernels in our seed. Let me see if we can see those chalkier looking colored seeds there. They're what we call opaque seeds. And they suddenly started to appear in very unexpectedly high frequencies in multiple populations. We have 2 papers out in Crop Science characterizing the quality differences that occurred. There were higher levels of trace elements and higher levels of essential amino acids. And particularly, the amino acids that we were very interested in were methionine and lysine. I don't know if you're familiar with it, but protein isn't just protein. And having a high protein corn isn't necessarily so great for nutrition. You want We want protein that that's very rich in the essential amino acids. Because if you don't have that sufficient quantities of those essential amino acids, the body can't utilize that protein to make new body. You have to have that right combination of essential amino acids. So we're really looking for the good quality protein, and that means the essential amino acids. And these things, these opaque kernels, had it. And so when we utilize those traits, and here we're looking at 3 conventional hybrids versus 6 of our hybrids, the averages, not only do we have more protein, and lysine and methionine and cysteine versus the conventional hybrids in the grain. And actually we had shifted the protein in the content of methionine, for example, in the protein. So we'd actually, per unit of protein, we had increased the methionine content about 22%. So it wasn't just that we had more protein, but we actually had changed the protein quality. Getting away from the proteins that make kernels hard, the alpha-zein in particular, and the gamma-zeins, which make that very hard, very dense, high test weight corn, shifting it more towards the beta and the delta-zeins and the non-zein proteins, which are softer proteins and have much higher nutritional value. So that's what happened. We also, working with Iowa State University, we developed a near-infrared calibration, which was very important for us to be able to routinely and cheaply measure the methionine content in our grain, and to be able to thereby breed for high methionine content. And this data right here shows from 2011 to 2016, 149 conventional, 1,250 of our hybrids from our yield trials where we measured quality. And you can see on average 16% more protein, 30% more methionine, and if we corrected for the protein level, 22% more methionine in the protein. Here's another example of it from 2015, 3 different trials with different types of our hybrids versus conventional hybrids. Mondamin by mondamin, mondamin by conventional, and conventional by conventional, where we took inbreds or mondamin inbreds or conventional inbreds from the companies, and we made crosses in between them, and we looked at them over that. And to summarize the information for this first set, which had 120 of our hybrids in it, 16.5% more protein. This one here with 90 of our hybrids, 15% more protein, 25% for this one here with 78 of our hybrids. Oil, 20% more, 13%, 11, 20, 19, and 20 in the lysine, and 24, 25, and 36 in the methionine. So what we've essentially done is we've bred for corn that has higher nutritional value. I apologize to the people on this side. It's very difficult with the split screen thing, so don't feel I'm ignoring you. I just I can only do so much. There's actually a value to this. If you look at methionine is a problem for organic poultry producers. Probably if you're an organic grain grower, probably most of the corn that you grow and sell, probably over half of that goes to feeding organic poultry. And methionine is limiting, as some of us here know, and it's been a big issue with the National Organic Standards Board, where we are allowing still the use of synthetic methionine. But that USDA has said that it's going to phase that out. And at this present moment, the poultry nutritionist that we're working with is saying that just the methionine in the corn makes the corn worth more, $1.37 a bushel more than normal corn. So that's because it replaces organic soybean meal or organic sunflower meal, which is also high in methionine. Also, we've selected, yeah?
Is there any value money-wise in the conventional market for higher methionine?
Not as much, because they can feed the synthetic methionine. Of course, if it goes for export, then perhaps there is a value, but for the domestic, it's not as much. I think everybody would like to see corn with higher methionine and lysine, but if they can supplement with synthetic amino acids, which are fairly cheap, it's not as big a deal. However, this is an issue for organic worldwide. It's not just in the States. The Europeans have problems with methionine. In Australia, they have problems with methionine. Worldwide, there's a real need for high methionine corn. We also selected for carotene in the kernels because carotene is important. A couple weeks ago, my wife and I bought $10 a dozen eggs, $10 a dozen, and when we opened them up, they were like super orange. They were incredibly orange, and that's what we've seen in Los Angeles and in Seattle, is that people are willing to pay for very expensive eggs if they can get that super orange color. And with our corn, we can feed a lot of it to the chicken. First off, we select it for high carotene content. We can feed a lot of it because we don't need to feed so much soybean meal. And possibly because of the soft kernels, we're just getting an awfully orange yolks. And here you can see some of our eggs and the insides of our animals when we gut them. They look like they're spray painted orange. And here you can see some of the eggs that my wife and I, when we get a little bit depressed how difficult it is to bring quality corn into the world, we just eat an egg. They're just incredible. These are some of our eggs, and these are from competitors. Actually, these are sold to the Japanese market for a very orange egg. But you can see that we're just off the charts in terms of the color thing for eggs. Now, I'm going to switch a little bit. Okay, so that's what happened when we selected and we did it. This emerging things happened with protein quality, and we were paying attention. Another thing that, Jeff, are you here somewhere? Jeff's not here. Anyway, one of the things that we noticed was that when we take conventional inbreds and we grow them under organic conditions, often they look really awful. They look like they're nitrogen deficient. And we call that you can call it cold turkey, or we call it culture shock. And after several generations of growing these things, often we see a difference. After we multiply them under organic conditions for several years, we see a shift, which has caused us really to wonder what's going on. I'll show you, we did a little research on it. And we took 4 different inbreds and 4 different breeding lines that had been raised conventionally and those that we had under organic management. And we grew them under organic management on our side in somewhat nitrogen-limiting conditions. And we found on average across those 8 lines that the ones that we had grown and, of course, inadvertently selected because they do better. You select the plants that do better, even though some of them were inbreds and supposed to be fixed. Nevertheless, we found across the board a 16% difference in chlorophyll. And also there were some differences as if the plants were becoming earlier under our conditions, which is as if they were shifting in their light response. So that suggests, well, chlorophyll is very much connected with nitrogen. So the more chlorophyll there is in the plant, the more nitrogen there is in the plant. And that suggested to us that the plants somehow would adapt to the conditions. And was this microbial? Were they fostering different types of microbes in their bodies? Was it genetic? Were they shifting in some sort of epigenetic way, or genetic way, their genome? Or was it both? We didn't really know what's going on here, but it led us to start thinking that, hey, these plants, they really adapt to their environments, to the organic environment, and we have to let this play out and get the best out of it. And that comes to the question of nitrogen fertilizers. When we began to see this, well, that plants really can adapt and become better, even when they're supposedly genetically fixed at getting the nitrogen, well, maybe we can do something to try to deal with this horrible nitrogen problem that we have on this continent. Nitrogen fertilizer, as you, I'm sure, all know, is polluting our well water and our surface water, something awful. And it's also polluting our air, as was referred to early on today. We got the big dead zone in the Gulf, which isn't getting any smaller. And it also, there's also side effects of it. For example, mosquito-borne diseases. are linked with all this nitrogenous water that's hanging around in our surface waters. And that, of course, can lead to human illnesses. So we need to do something about that. One of the things in the previous presentation here, we were looking at rooting systems of our corn and conventional corn or conventional varieties, and it appeared that our hybrids might be having very dense rooting systems with lots of roots on them. And that seems to be what we're seeing if we look at a set of conventional inbreds under nitrogen deficient or nitrogen limited conditions. If we look at this is LH206, LH123, and these are two from a commercial company. They tend to have very vertical roots and not to be so very densely branched, whereas our inbreds tend to be very densely branched, particularly in the topsoil, and to encompass a great volume of soil. So the rooting system seems to be adapting to these, when we select under these low nitrogen conditions that we select under, seems to be adapting by becoming branchier. Now, I want to say a word here about nitrogen in corn and what can we do with breeding and besides breeding on the whole nitrogen issue within the organic situation. I want to step back just a moment and talk a little bit about the whole issue of soil quality. And this slide here shows a picture of corn following, or well, the corn has not been planted yet. This is actually, we had oats last year, and we've just planted, we're just about to plant the corn. This is sweet clover was seeded together with the oats, and we turned the sweet clover under, so we had a green manure effect. And wow, look at that soil. It's one of those soils that you can really stick your hand in. And if you look at that soil and you look at this picture here, this is a plot right next to it, and this corn had sweet clover the previous year. It had oats and sweet clover, and then we grew corn, and now this is after the corn. And so I just want you to ponder this for a moment. If you look at these two pictures here, you can see all these crumbs. You see all this structure in between maybe a quarter of a millimeter in size or an eighth of a millimeter, and maybe 8 millimeters in size. You see all these this crumbliness that happened by this feeding the soil. And this feeding of the soil somehow disappeared, or the marks of the feeding of the soil disappeared when we grew the corn. And I don't know if you've noticed it, but I've noticed this now for decades. Corn makes the soil hard. The Europeans noticed this. They called corn the syphilis of civilization. But you know what? There's no other crop like corn, I mean, in terms of what it can produce. So it takes a lot, but it gives a lot. And if we want it to give a lot, we know it takes a lot. And if we're doing this thing organically, we really have to be fostering this side, all this soil making and not the soil taking. The corn is a soil taker. The cover crops are soil makers. But within that situation, what can we do in order to more efficiently utilize the nitrogen? I mean, here what's happened is all this young organic matter from that decomposing green manure has coated the sides of those little peds, those little crumbs, and made the soil so that when you break it, it's all crumbly, it's not jagged and rocky looking or mineral looking. What can we do to even get to really utilize that resource, the nitrogenous proteinaceous substances that are coating those little peds? Here you can see it with vetch. Isn't it probably a pretty bad picture, but these little crumbs here you can see are just getting cemented together by all this young organic exudates that are coming out of these vetch roots. Here we see actually a longer-term experiment that we had, a 6-year experiment comparing a corn and soybean conventional system with a 6-year system under organic and biodynamic systems. Biodynamic systems, we use certain bio sprays that were to stimulate the plant. And here what I want to show you is the soil structure, the big blocky crumbs that were, or blocky pieces or massive pieces of the soil that were there, increased in the corn-soybean system relative to the system as the 6-year system where we had 2 years of it was in perennial forages. So having that perennial forage in the system really did increase the crumbliness of the soil, and you can see it. So you can actually read your soils and see, you know, the condition of how much is your soil going to be able to get out of the soil organic matter to grow the crop, just by looking at the soil. So I encourage you to look. And bio sprays, if we look at these experiments, they can also be useful. What they seem to be doing is stimulating root growth, and the stimulating of the root growth versus not using this is 2 years of data on roots. It can be important for stress resistance. If you have a stronger rooting system and you have stress, that can help you to avoid the stress and to still get a good yield. And here you can see we're using an open pollinated corn variety for this, where we're using these sprays. And on the bad years, that's where we got the yield increase of using these sprays. And on the really good yielding years for those particular variety, then we didn't see any difference. So the roots are a kind of insurance. And so as we move along and select plants for their value for organic farming situation, perhaps it isn't a surprise that we're getting more roots. It's the same situation with wheat, that the yield increase here is great at the low yielding years, but in the very high yielding years, then we don't even see maybe even a slight yield decrease. So the strategy of investing more in roots or stimulating the plants so that they have more roots, or breeding plants to have more roots, well, there's costs for doing so. And those costs will probably pay off on the tougher years, and they may not pay off in the super nice years. So I think that's the way I'm thinking about this data at this point. In terms of working with the beneficial bacteria, or starting to work with beneficial bacteria, that's perhaps another way that we can optimize nitrogen. And here, there's different things that bacteria can do that are useful. I'm going to focus on the nitrogen fixation angle here. This is a kind of a literature survey of research that's been done on whether corn can or cannot fix nitrogen. It's a little bit dated here, but here you can see in the yellow, you see the trials where they really didn't see any effect in the literature, or any sort of clear indication that any sort of sizable nitrogen fixation could occur in corn. But on these other ones, almost in all cases, there was an effect of the variety. So it made a difference which variety you choose in terms of whether or not it actually would respond to bacteria by fixing any sort of nitrogen. Getting into this thing, there's a kind of an interaction that happens when a plant does partner with a microbe and fix nitrogen. There's a kind of a modulation that goes on. The plant down modulates its defenses. It has to feed the microbe. The plant, there's a kind of courting that goes on. There's signaling between the bacteria and the plant. So there's a little dance that goes on between the bacteria and the corn. And not all hybrids can do that. So that's I think the situation. The corn, corn in general, but also with different varieties, when it's exposed to conditions where it doesn't have enough nitrogen, it will change its relationship with bacteria. It makes a more inviting environment for them. One thing it does is it downregulates the amount of the ethylene that it produces, which is an inhibitor for bacterial growth. And it also produces aerenchyma. And I don't know if you all know what aerenchyma are, but the plant actually bores out its root, and it makes these little tubes in the root. I'll show it to you in just a minute. And those can be sites where nitrogen fixation can occur. Also, it may be that fusarium. I think many of you who produce corn have heard of fusarium. Fusarium is a big problem for corn production worldwide. And fusarium is a very dominant microbe. It's a fungus. It lives also inside the plant, and it's transferred from seed to seed. It's what's called an endophyte. Once it's established in the plant, it tends to dominate other microbes, other fungi, and also beneficial bacteria, in particular some of the bacterial classes that can fix nitrogen. And unfortunately, with our breeding programs, I can make an argument that there's certainly literature supporting it, that we have inadvertently selected plant for partnerships with Fusarium. That's our dominant endophyte in our current varieties. And we didn't know that we did it. There were things that we selected for, such as first-generation corn borer resistance. The plant produces a biotoxin, which Fusarium loves and seems to thrive under those conditions. So by selecting for first-generation corn borer resistance, we selected for Fusarium. For selecting for very stiff stalks, we selected for Fusarium, because Fusarium induces a plant to form stiff stalks. And there's other things, a small tassel, the lack of tillering, resistance to smut. All those characteristics favor Fusarium and Fusarium's activities in the plant, heat stress resistance, and even we've found a certain amount of emergence and seedling survival. Probably all relate to beneficial effects of Fusarium in the plant. Now there's other ways in which nitrogen can be fixed. There's Mexican corn can develop exaggerated brace roots, and they can be covered with a mucigel, which will favor nitrogen-fixing bacteria. The recent paper published by UC Davis and University of Wisconsin has illustrated that. And we found also in Wisconsin, in working with those Mexican corns, that the corn seems to actually oxidize or have a system for eating the tips of its roots, which we think is colonizing the roots and digesting them. And I'll show you pictures of that in just a sec. First, the aerenchyma. You can see this is a cross-section of a root bored out by the plant itself. Beautiful, huh? Aren't they artists? And they do it with hydrogen peroxide and other peroxides. And in there you can see a photograph showing the location of a nitrogen-fixing bacteria living within those tubes. Now let's go to the prop roots with the mucus on there that fixes the nitrogen. That was discovered by this gentleman here, Thomas Boone Hallberg, in Oaxaca, Mexico. And he told all sorts of people about it, including myself. And there was some very good research that was done on that by this gentleman here, Ronald Ferreira Cerrado, identifying the nitrogen-fixing bacteria on those brace roots. And then later by Paulina Estrada de los Santos and her team identifying some of the microbes that live in that mucus. And so there's been some really good work done already by Mexican workers that people just ignored. But now UC Davis has come out with something it's really pretty hot. So people are really thinking about it, and they've done some real good work at UC Davis, proving that nitrogen fixation can occur in this land race. I believe it goes all the way up to 80% of the total nitrogen in the plant, depending on the year and the environment and the method of analysis, because there's no perfect way of estimating nitrogen fixation in corn. But anyway, quite a bit can be fixed, and that this particular exudate from these brace roots is highly conducive to nitrogen fixation because certain bacteria are cultured by it. So it's some great work. In Wisconsin, we've been breeding it, and here you can see what I was talking about, the exudate. You can see that little drop there, and then you can see that little black thing in there where the roots actually end up looking like they've been burned on the tips, almost like cigarette butts at the end of the growing season when the plant is senescing. And here you can see several generations of breeding these things for Midwestern conditions, and for 7 generations of breeding to be able to fix it into varieties that we can grow here. That's kind of what they look like when we got started with some of them. And going back in history, I can trace it back to 2009, where we found we had a site with very low nitrogen, and we had a bunch of different conventional inbreds, and we had these land races of corn from different parts of the world, and we inoculated everything with nitrogen-fixing bacteria, and wow, some of these land races look like they've been heavily fertilized with nitrogen, whereas the conventional inbreds, this is B73, a very common inbred, used in most of the hybrids, its descendants in the Midwest. And let's see, go back. And when we looked at the isotope ratio between N14 and N15, there's more N14 in the air and more N15 in soil, relatively speaking. The balance shifts to be more in the air, and we look at the ratio in the plant, and we can estimate nitrogen fixation from that. We had some of these land races here that appeared to be fixing almost half of the nitrogen from the air. Wow, that was exciting. So what we started to do was to breed with that, and we made crosses with it. And the first year we looked at those hybrids versus in the front you have some conventional. These two are conventional hybrids. The averages for conventional hybrids where we had a control, and then we inoculated with nitrogen-fixing bacteria. No response. But these hybrids that we'd made with these putative nitrogen-fixing land races, they did quite well. There was a response, a grain yield statistically significant for protein yield and the yield of the essential amino acids. So more nitrogen was being harvested by these plants when they were inoculated than when they weren't inoculated with nitrogen-fixing bacteria. And then we started doing really crazy stuff. We started selecting. Okay. Well, before I should go into that, let me tell you. Here you can see something very interesting. This is a yield of the conventional organic hybrids, and this is a yield of these new hybrids. And this is the protein yield, and this is the grain yield. And here you can see that for every unit of yield, you're getting more protein for these particular hybrids. And so that's the point. That actually, and this is carried down whenever we have comparable yields, we're having much higher protein yields per acre. I think someone was talking about what kind of new metrics can we use for the type of corn that we're breeding. How about protein per acre? So the crazy stuff. We started growing plants in an old quarry just to see who would survive. And the soil had practically no nitrogen in it. And what we did find was that there were certain families that at least made it up to flowering. And that was impressive. Of course, there's some rain coming in, there's some rainwater coming with nitrogen and so on, but nevertheless, most of the plants died in the seedling stage. So and those were the ones, of course, that we continued working with. And this shows we switched to a 1% organic matter soil, a very sandy soil, well stocked in nutrients otherwise, but not nitrogen. And here you can see our check really crashed, a population, a corn belt population. This particular line here, the C line, that's a Mexican, derived from a Mexican land race, did really well, relatively speaking. And where we've really found the difference, and we looked at the normal corn belts, was they went barren. And then we had these particular lines from different sources that would put on more ears. So they seemed to be able to do something under those low nitrogen conditions. By 2015, we continued breeding, and we thought we fixed the trait in an inbred, the C46, which was referred to in the last presentation. And you can see it's a very droopy, dark-leafed thing. Wherever we grew it, we didn't have to inoculate it in order to get that. It appeared really dark green wherever we grew it under nitrogen-limited conditions. It looked convincingly nitrogen efficient. The year before, we had tested the protein content in the leaves. It was very high. So it had a lot of nitrogen in it. Here you can see it the next year. a whole set of them on this side, whereas sister lines up here that don't seem to have the trait on the other side. They look like they have more nitrogen. They're taller growing. They just look like they have more nitrogen. In 2016, we tested a whole bunch of different hybrids, 3 different sets with some overlap, 125 here, 150 here. We tested a whole lot, but this data just shows the top ones. Our top 5, the conventional high 4 hybrids that we had. And this was on a low fertilization site, a low fertility site, actually a site that had a poor soil quality and was in conversion to organic. And here's another one of those sites. And here we had an old organic site, which had excellent soil fertility and about 50 tons of manure per acre. So it was just really heavily manured. And here we didn't really see much difference in yield. In protein, not that much difference either. The protein was high, but super fertilized. But on the lower fertility sites, that's where we saw differences in yield and much bigger differences in protein content. So these varieties seem to really be responding under conditions where nitrogen was limited, was our impression. I think, let's see, how are we doing for time? This is a little bit confusing here. We got 15 minutes? Okay, all right. Color differences. In general, we're looking for, when we look at the inbreds, we're seeing about, when we do chlorophyll ratings, we're doing about 15% more to maybe a third more at the top, depending on the inbred that we've developed, than conventional inbreds from the companies. So they look like they're getting more nitrogen. And here you can see in our field strip trials this year that we did together with U of I, these 2 were the cultivar, which was the very dark one here is the one that we think it's C by C, essentially. It's 2 different lines derived from that same big ear that I showed you on that 1%, the C family. And they seem to make a good hybrid. But unfortunately, they seem also to be susceptible to gray leaf spot, which we didn't know. We wouldn't know until we took it out, from one year to the next. Sometimes you don't know if you have a bad disease thing. So they didn't yield as well as we would like. Though they look great, they looked really dark green early on. There they are on the Adsit farm where we had our field day this year. In terms of the chlorophyll scores, they were the highest. But you can also see they developed a gray leaf spot later on in the season, and they didn't do as well. And I think they didn't do as well up here also as the other variety that you were, 2 varieties.
Keep in mind, everything did that gray leaf spot this year in the conventional world. I mean, up to 15 birds in the trial, most of them we tested. So,
Oh, is that so?
yeah.
Oh, you comfort my heart. Yeah, it was the worst I've ever seen. It was unbelievable. Yeah.
As far as competing in the real world, there's not that much resistance out there.
Okay, great. We did find lines that are resistant. See these ones here, and even tar spot. They seem to be resistant to tar spot too, so that was wonderful. Yeah, and so, and if we look at our yields over 13 different sites, this was our top hybrid, which I think was also, that slide went by really fast, but I'm pretty sure that was the top yielder down in Illinois also. Anyway, and the Illinois data is not included. Over all the sites, we actually did better than the conventional checks, but we were higher in moisture because some of our checks were earlier. When we compared ourselves with FOSS 8507, which is a really good hybrid in terms of yield, we yielded, I think it was 11% less. So, we're going to need that if that's the hybrid of choice, we're going to need the premium to have the same economics. The other one, which did well up here, 17 by C46, which Martin was just talking about, yeah, on average we did better than the Czechs, and the moisture was the same. But against the FOSS 8507, we didn't do as well. And yeah, okay, well, we learned from that and we continue. But I think we're getting there, I guess is the point in terms of competitivity with yields. And this is just 1 year's worth of data anyway. So FOSS 8507 actually, not in 2019, but 2018, it kind of crashed for us on some sites. So the racehorses may not always do well under stress conditions. And we are breeding kind of, I think what we're breeding, maybe not so much racehorses as workhorses a little bit in terms of being able to take the stress and the lower input conditions that I think actually we need to be breeding for, because we just got to stop using so much fertilizer. Yeah. Flowering date, we range. We say that this one here is I believe 106 day. I have to check on that one, and this one here is later. This one I think we were talking about 112 day in terms of relative maturity. Okay, so where do we expect this thing to be able to work? And this is something because now we started working with some conventional farmers, not just organic, and of course we're working together with U of I, and if you're interested in this stuff, talk with Carmen and Michelle and Martin about participating. But first off, one of the big questions is Roundup. It turns out that a lot of farmers don't know how to farm without using Roundup. And the problem is that, I think that these varieties are going to do really well where we have good quality soils, good quality structure, root disease is suppressed, not that much nitrogen, the plants have to work to get it. Maybe we use some biostimulants, but I don't know that we're going to do so well under conditions where there's just an awful, and be able to reduce the nitrogen under conditions where fusarium is dominant. And fusarium, I believe, is dominant in our conventional systems. If plants are being challenged by a lot of root rot, they're going to be turning on their defense systems. Who wants to have partnerships with beneficial organisms when you've got all your defenses up? Soil structure is poor. And you saw that what happened. I've demonstrated to you that soil structure goes bad under corn-soybean system, goes bad after corn. If you're not maintaining your soil structure, it's going to be harder for the roots to stay healthy. There's going to be more conducive for disease because of the residues of corn from previous years or fusarium in that, growing in that. So, and then the Roundup, which also is an issue. And I say that because we did this survey years ago when I was working with Michelle before. And we found when we did this survey a lot more root rot on the conventional systems versus the organic systems. This was a survey we did. There's a number of farms we looked at. And here's the difference in the root rot. And it was especially an issue where corn followed after corn. We had up to 30% root rot when the plants were flowering. So it was an issue. And you don't often see it because you don't dig down there and look, but there is considerable rot down there, and particularly where corn follows after corn. And Roundup, unfortunately, there's evidence out there now that Roundup tends to enhance fusarium colonization of roots. And partially that seems to be due to the fact that it affects organisms that suppress fusarium, such as pseudomonads, beneficial pseudomonads, the good bacteria, and then the fusarium gets out of control, grows on the roots, grows inside the plants, and then results in toxins. And the fusarium has just got an awful lot of toxins in it. It's really terrible stuff. And I'm beginning to come to the realization that we've got to do something about this in the organic movement. We have to breed corn that is really fusarium resistant. It's a hidden issue. It lives in the seed. It's transferred from one generation to the other. It produces these horrible toxins if the weather isn't right. And they're just poisons. They leak into our food system. They do get into milk. They've been detected in mother's milk, going into babies. You know, even though they are diluted, they are nevertheless an issue. And this shows some of the levels that some of the conventional farmers are. This is from one conventional farm we got some data from, the levels where they're experiencing hemorrhagic bowel syndrome in their dairy cows. And this is the levels of some of these toxins that they're seeing on these farms, and they're really high. And that coupled with Roundup on the plants, animals eating that, may just be causing some of these bowel problems. So I think we need to move in the other direction. If you think of healthy soils having a corollary to our digestive tract, and then this is showing us that we're the direction we don't want to move in, and how we actually have to move the whole system out, and that the breeding, what we're doing for this nitrogen-efficient corn, has a place in a healthy system. And that's I think, what I wanted to show you today. Do we have time for a couple questions? Yeah. Or discussion?
Either or.
Do you know anything about the relationship between the organisms that are in the roots of corn and the organisms that are in the roots of alfalfa?
I do know there is evidence out there, but it's old stuff, that part of there is a group of scientists who believe that part of the problem that we have with red clover as a green manure has to do with endophytes that live in the red clover that somehow colonize young corn seedlings and have a negative effect on them. And I don't know if that's true or not, but in that case it was a negative relationship. In general, I think the problems between legume residues and corn, sure, legumes can give a lot of nitrogen. They do a fantastic effect on the soil, but you do have to be careful, as we were talking about in a previous session, that you allow those residues to decompose long enough in the soil before you plant your corn, because you can stunt your corn. The corn can do very poorly if those residues haven't been digested by the soil. And
Ties up the nitrogen.
Fusarium has the impact on the plant that the plant appears to be nitrogen deficient. So some of these microbial infections can mimic or can induce nitrogen deficiencies in the plant, for example.
When do you think any of these nitrogen-fixing corn things could be commercially available?
So we have been trying them in strip trials now. We're multiplying seed in Puerto Rico this year, and we made some hybrids, so we're doing more strip trials next year. And I'm hoping this year we're going to be making hybrid seed for larger scale plantings, but still not huge. We're sort of creeping out with it and bringing it out. I was very encouraged to see the results that, for the first time, that we had down here in Illinois. That was an enormous relief, Martin. You never know how your stuff is going to do until so thank you. So it looked good down here, and yeah, you know, hey, that protein quality, that carotene, you know, we got really nice quality there, and I think a lot of people who feed livestock are going to want that to feed their animals. So yeah, we're going for it as fast as we can with Foundation Organic Seed are the folks that have stood behind us on this. Everybody's had an opportunity. I've talked with all the companies, but the one that stood behind it was Foundation Organic Seed. We're looking to try to do a kind of an association with the seed, so that people who have to join the association, by buying the seed, they become members of the association. They make an agreement. When they buy the seed, they won't give the seed away, because we're not patenting the seed.
But it's hybrid though.
Well, we're not patenting any of our seed, whether it's inbred or hybrid or our wheat varieties also. We're not patenting it, but we want to form an association of people who agree to protect the seed And we want also, it's just a starvation act to try to get breeding done publicly anymore. It's just living from, it's really tough. And we need a stream of income to pay the bills. And so what we've been talking with is with the end users, the people who sell product, who sell food product or sell feed. And we would like to get a certain percentage of their sales going back to the breeding program. And then they can use, we're going for a trademark and a logo that, you know, this is bred a certain way. We're going to register our varieties on our website, and we're going to make it public. So what we're trying to do is a system, anybody can join it, but they have to obey the rules, and there has to be an income coming back to the breeder from the end user.
What about giving the inbred lines to a farmer and let him generate his own hybrid?
That's fine with us. Anybody can make their own hybrids. That's just fine. That's yeah. So if you, yeah.
Any other questions?
Just one more remark. We do have a wheat variety that we're bringing out in the same way. It's an old wheat variety that's a sort of a free threshing spelt, and we're working with farmers and millers to try to get that out the same way with a kind of an association. Great.
Thank you, Dr. Goldstein.